For most plants, the primary site of photosynthesis are leaves. The leaves are orientated into a flattened structure to maximize surface area to capture the most amount of sunlight possible. In most plants, they acquire water from a transpiration pool, which is essential for the water required for photosynthesis. The internal leaf structures are set up in a way to facilitate maximum capture of sunlight and minimize water loss.
The internal structure has many layers that contribute to the overall functionality and operation of the leaf. The waxy cuticle is the top layer of the leaf. It acts as a waterproof layer to minimize water loss. Its functionality comes from it being hydrophobic and not absorbing water into the leaf, forming water droplets. The next layer down is the upper epidermis. It contains no chloroplasts and allows for light to enter while preventing water loss. Following this is the palisade mesophyll. This is the site of photosynthesis and hence is located on the upper surface of the leaf. It is a tightly packed layer of chloroplasts and its cells have a large vacuole for water storage. The spongy mesophyll is the main site of gas exchange, hence is located on the lower surface of the leaf. It contains an intercellular space which usually contains water and oxygen. They also have a small amount of chloroplasts, which means they can conduct a small amount of photosynthesis in comparison to the palisade mesophyll layer. The central layer is the vascular bundles, which contains the xylem and phloem. The outside coating of the vascular bundle is made up of a large group of closely packed cells called bundle sheath cells, where the xylem and phloem resides. The xylem is the vascular system that functions to facilitate the movement of water and minerals from the roots of a plant to nourish the leaves. It is composed of dead cells that are hollow to allow for the passive movement of water. This comes to an advantage as water is not required to rely on cells to move it around. The phloem is a living vascular system with the purpose of carrying sugar and organic compounds throughout the plant. The next layer is the lower epidermis. It functions as the upper epidermis except it is on the lower part of the leaf. Following this is another waxy cuticle, which is just like the top waxy cuticle and helps to prevent water loss. The stomata is a small opening between the guard cells, responsible for gas exchange, where it allows carbon dioxide to flow in and water vapor and oxygen vapor to flow out. The transpiration rate is regulated by the opening and closing of the stomata. The guard cells are the two cells surrounding the stomata to control the rates of gas and water exchange. Guard cells flank the stomata and can occlude the opening by becoming increasingly flaccid in response to cellular signals. This is particularly important as in dry periods where conservation of water is vital, these stomata can close and limit the amount of water lost.
Transpiration brings water upwards from the root to the leaves, which is known as the transpiration pool and is essential for photosynthesis. It begins with the evaporation of water from cells inside the leaf where water vapor diffuses out of the stomata. The evaporation of water through the leaves causes water to be drawn in from the roots by osmosis to replace the water that has been lost. The rate of water uptake is regulated by special water channels, known as aquaporanes, on the root cell membrane. Transpiration rates are affected with higher temperatures, increase in humidity, and increase in light intensity and wind exposure. This in turn affects the rate of photosynthesis. Xerophytes are plants that can tolerate dry conditions due to the presence of reduced leaves, thick waxy cuticles, and low growth to combat the higher rates of evaporation.
Plants perform photosynthesis as it generates the food and energy they need for growth and cellular respiration. The significance of the role of plants in supporting the environment is evident in observing the functionality and processes taken by plants through the analysis of the internal leaf structure and the process of transpiration. The leaf is structured in a way the plant can carry out photosynthesis and transpiration efficiently.